Good evening. I'm Barbara Mead. I'm one of the founders of Politics and Prose, and I'm so happy to see we've got a good crowd here tonight. Now, listen, I've got somebody for you really to clap for, and that is it for David, because this is opening night for David. This is the first stop on his book tour. And so it's a real pleasure for me and by the time that he gets back to Bozeman, Montana, I hope that he'll have some good tales to tell about politics and prose, right, David? And David has come to talk about his new book, The Tangled Tree. I was telling him this past Christmas, I gave to people in my family, I gave a handful of DNA kits. And because I thought we could get the definitive word about where that everybody in our family came from. So everybody sent some saliva off to the respective, I got several different kinds, to the respective addresses. And they came back, and now we all thought that we knew exactly what our DNA, our heritage is, or what our family tree looks like. And now comes along David with his new book to say, no, that's not true. It's much more complicated than that. You have no idea about horizontal gene transfer. Ancestry.com never told us about horizontal gene transfer. David has described his youth as having four great influences. First was his father, who led him far beyond the city limits of Cincinnati on wilderness paths that went into forests, along rivers, and up mountains. Last night, I streamed on TV a movie called Walking Out, which is based on a short story that David wrote 40 years ago. Actually, the story exists in an anthology that was put together by Raymond Carver called Short Story Masterpieces, something like that. But this movie was just recently made. It was featured at the Sundance Film Festival, and watch it, you'll like it. But it features a estranged father and his teenage son, and they're going on a hunting trip through mountains and forests. And I couldn't help but thinking about how that David had described his youth with his father, except he was not estranged from his father. So it was... how did I get on the subject? Oh, I know that it was his father was his first influence in his great influence in his life. He says he was also influenced by three great teachers, which included two Jesuit English teachers in high school in Cincinnati, and then when he was an undergraduate at Yale, the novelist and the poet Robert Penn Warren, who acted as both a mentor and a friend in helping him write his first book, which was a novel. After Yale, he received a Rhodes Scholarship to go to Oxford University. Then, at some point along the way, I think it was earlier than Oxford, he became obsessed with the novels of William Faulkner, which it led him to, when he started writing, to be a fiction writer. Walking Out came from that period. But he soon learned that you got more money from writing nonfiction than you got from writing fiction, and so he kind of switched over to the other field. Out of this life progression, I emerged a man I think of David as a human glass prism, that his life experience is refracted into a wide dispersal of light waves in different directions and in different colors. In his 30-year career, David has written 15 books, fiction and non-fiction, as well as several hundred pieces for magazines and newspapers, including National Geographic, for which he bears the title Contributing Writer. And it was under that title that he is the only writer to have written the entire text for a single issue. And that was the May 2016 edition, which was devoted entirely to the Yellowstone Park ecosystem. For these accomplishments, David has been honored many, many times, including an award from the American Academy of Arts and Letters, the Lannan Literary Award, as well as the National Association of Science Writers Award. And he has won the National Magazine Award 3 times. The New York Times calls him our greatest living chronicler of the natural world. His last book, Spillover, which I introduced here 6 years ago, was shortlisted for 8 national and international awards, including a finalist for the National Book Critics Award. And it won 3 of the 8 it was a finalist for. Now, if you look at the back of your book, you'll see Elizabeth Colbert saying, "No one writes better about complex science than David Quammen." And I think that probably when you leave here this evening, you'll think nobody speaks better about complex science than David Quammen. So here's David to tell us all about the Tangled Tree.
Barbara, thank you so much. Thank you so. Do you want to go on tour with me? You could introduce me maybe every night. I would like that. Thank you all for being here. This will be kind of a relaxed and informal. I hope in the second half of it will be conversation back and forth. And I'm going to just make a couple of tweaks, additions to what Barbara said, and then I'll talk about the book. First of all, my branch of the Tree of Life looks like this section of the audience right here. That's what my branch of the Tree of Life looks like. And it's great to have family here. It's great to have all of you here. I would also add that among my 4 or 5 greatest influences, Barbara mentioned my father, but also my mother. If I have any sort of a caustic sense of Irish humor, it's thanks to Mary Louise Egan, for the record. And finally, yes, I did discover that nonfiction pays better than fiction, but that wasn't the only reason that I switched, Barbara. There was more. There were some other considerations. Okay, so speaking of nonfiction, books, fiction, how many people here have ever written a book? Already a serious. How many people here have ever thought about writing a book or written a book? Hands all up together. Okay, I take that to be just about half of the audience, and that doesn't surprise me. I've seen and heard that elsewhere too. A lot of people think I should write a book. I will write a book. Many people do write a book. I want to give you, just in relation to this project, a little bit of hard-won counsel on that if you ever decide to write a book or another book. When you pitch it to your editor or your potential editor, don't walk in and say, "This will be a book about the history of molecular phylogenetics." Okay? Don't do that. Don't even go in and say, "Oh, but no, this will be the lighter side of molecular phylogenetics." Don't do that. And if you add, and it'll include a lot of really fascinating things like horizontal gene transfer and endosymbiosis and the origins of the eukaryotic cell, at that point you'll see your potential editor starting to maybe glance toward the door or doze off or whatever. So, what do? If you're interested in the things that I was interested in 5 years ago, you say, this is going to be a book about the most important biologist of the 20th century that you've never heard of. And his name is Carl Woese. And he was a little white-haired man in Urbana, Illinois, who made a discovery in 1977 that changed the way we understand the history of life on the planet. Okay? So that's what this book is about. It changed the way we see the tree of life being the classic Darwinian figure of representing the history of life on Earth with a single great trunk and a few major limbs coming off, and then branches and divergence and more branches and tinier branches and more divergence and twigs. It changed that picture. And the people who followed this man, Carl R. Woese, continued to change the picture even more, to the point where our sense, our image of the history of life on Earth is not a tree anymore. It's something else. It's tangled. It's a tangled tree. It's a web. It's a network. It's not just divergence, representing the divergence of lineages that create the great canopy of leaves that represent living biological diversity on Earth. We now know that there have been limbs not just diverging, but converging, limbs that grow into other limbs and branches that have moved sideways from one part of the tree into another part of the tree. That's this phenomenon that the scientists call horizontal gene transfer. This project began for me in I'm going to watch my time to make sure I don't just rattle on and we leave plenty of time for conversation. This project began for me in just about 5 years ago, maybe 5.5 years ago, let's say spring of 2013. I had finished the book Spillover that Barbara mentioned, a book about emerging diseases that come from animals and get into humans, Ebola and a number of other scary viruses and other pathogens that pass from animals into humans. So I was finished with that and finished with supporting that and book touring for that. And the 2014 Ebola outbreak in West Africa hadn't started yet. That's another whole story that we could talk about. And I just was reading widely and thinking about what's the next book project for me. And I happened to come across in some journal article a mention of horizontal gene transfer, HGT, horizontal gene transfer. And first I thought, horizontal gene transfer, what's that? That genes moving sideways across species boundaries? Well, that's not possible. That we know. Species are defined by the fact that genes don't move sideways. Genes move vertically, not horizontally. Genes move from parents to offspring. And even if you're not a complex animal or plant with parents, a species of animal and plant where there are parents and there's sexual reproduction and there's offspring, still genes move vertically. Even if it's a bacterium reproducing itself by fission, genes move vertically, don't they? And I started reading about this, and I found more journal articles and then more journal articles, and then I discovered a man named Ford Doolittle, an American who has been in Halifax, Nova Scotia for the last 45 years or so, teaching at Dalhousie University, and has written a lot from the point of view of a scientist, of a microbiologist and a molecular biologist, about horizontal gene transfer. He wrote some very important articles, one in Science in 1999 and another in Scientific American in the year 2000. So I read those by Ford Doolittle. I thought, wow, I had no idea. This is bizarre. It's supposed to be impossible. And then I read some more, and I discovered this man, Carl Woese, who was dead by that point, died in December of 2012. And so I started working backward and working outward and reading about Carl Woese, and found one very good book for sort of an academic audience on all this, and that led me to some other journal articles and some other scientists. Anyway, I did what you do if you're a nonfiction writer, which is follow these threads. And it became for me, because Woese was dead and because he was the mysterious, crotchety figure in the middle of this whole story, Woese became my Citizen Kane. Anybody who hasn't seen the movie Citizen Kane, please raise your hand. Okay, don't feel bad, but see it. See it. And you'll understand what I'm talking about. Woese became my Citizen Kane, and I was the faceless newsreel reporter who was sent out by his editor after this great legendary figure died. Charles Foster Kane fell over dead, and his last word was Rosebud, and nobody knew, well, what the heck did that mean? What, Rosebud? Rosebud? Whoa. Is that the explanation of his life? What's the story of this strange, difficult, many-sided man? So Carl Woese became my Citizen Kane, and I was the newsreel reporter going to his former graduate students and his former technical assistants and his colleagues and his friends and his sister and a number of other people, saying, who was Carl Woese? What was the deal with Carl Woese? What did his Rosebud mean? And that's really the essence of this book. It's about the tree of life, the radically redrawn tree of life, in light of discoveries from genome sequencing, the sequencing of DNA and also of another nucleic acid, RNA, a type of information molecule. Everybody's heard of RNA, right? Even if we don't know exactly what it does. You'll get a big dose of RNA in this book, but clearly explained, I promise you. Discoveries from genome sequencing that began with Carl Woese using very primitive methods at his lab in Urbana, Illinois in the early 1970s, using toxic chemicals and high voltage and explosive solvents and radioactive phosphorus in order to cut apart long molecules, DNA and RNA, from different forms of life and sequence them, try and learn what the sequence was of the letters that compose those molecules. The letters A, C, T, and G compose all DNA molecules. The letters A, C, G, and U compose all RNA molecules. And genetic sequencing is finding out how those four letters figure, recur, repeat, are varied in a long, long, long molecule. And nowadays, you can get your genome sequenced for what? $150? Maybe a little bit more for the full genome. An undergraduate using a fancy machine can sequence a genome in an afternoon, doing what it took Carl Woese a year of dealing with these difficult methods and toxic chemicals to do. Actually, and Carl Woese could only do fragments, small fragments of a genome in a year. But he did that. He patched together this difficult methodology. He crawled on his hands and knees across the Bonneville salt flats of genome sequencing in its early phase to get a few bits of data and compare different creatures with one another. And one of the things that he did, his earliest important discovery, was on the front page of the New York Times, November 3rd, 1977. Doing this, he had discovered a completely new form of life, a third form of life. Until that point, we thought there were two major limbs on the tree of life. There was bacteria, which were small creatures, one cell, no cell nucleus, very simple. And there was everything else. The fancy name is eukaryotes, but what it means is animals, plants, fungi, and every other kind of creature that's composed of complex cells that do have a cell nucleus and complex internal structure. So 2 forms of life, bacteria and everything else. November 3rd, 1977, Woese announces, no, there's a 3rd form. There's a 3rd major branch on the tree of life, and it became known as the Archaea, because it seemed old. It seemed like these creatures were very old and primitive. They were creatures that looked like bacteria through a microscope and had been mistaken for bacteria for hundreds of years, since the time of Leeuwenhoek, when people started looking at things through microscopes. But looking at their DNA and their RNA, looking at their genetic sequences, Woese saw that these things, these little bugs, although they're single-celled, and although they don't have cell nuclei, are more similar to humans than they are to bacteria. That's what he discovered at the genetic level, at the DNA level. And that was on the front page of the New York Times. And that was Carl Woese's 15 minutes of Andy Warhol-style fame, and then he disappeared back into his laboratory for the next 40 years, from which I've tried to excavate him in this book. I want to read a little bit of it. I'll read a couple of paragraphs. First, I'll read just a little bit about Woese. This is in the very early part of the book when I'm just sort of setting the scene. Carl Woese was a complicated man, fiercely dedicated and very private, who seized upon deep questions, cobbled together ingenious techniques to pursue those questions, flouted some of the rules of scientific decorum, made enemies, ignored niceties, said what he thought, focused obsessively on his own research program to the exclusion of most other concerns, and turned up at least 1 or 2 discoveries that shook the pillars of biological thought. I just mentioned one. To his close friends, he was an easy, funny guy, caustic but wry, with a love for jazz, a taste for beer and scotch, and an amateurish facility on piano. To his grad students and postdoctoral fellows and laboratory assistants, most of them, he was a good boss and an inspirational mentor, sometimes, but not always, generous, wise, and caring. As a teacher in the narrow sense, a professor of microbiology at the University of Illinois, he was almost non-existent as far as undergraduates were concerned. He didn't stand before large banks of eager, clueless students, patiently explaining the ABCs of bacteria. Lecturing wasn't his strength or his interest, and he lacked eloquent forcefulness even when presenting his work at scientific meetings. He didn't like meetings. He didn't like travel. He didn't create a joyous collegial culture within his lab, hosting seminars and Christmas parties to be captured in group photos, as many senior scientists do. He had his chosen young friends, and some of them remember good times, laughter, beery barbecues in the Woese home, just a short walk from the university campus. But those friends were the select few who somehow, by charm or by luck, had gotten through his shell. That's a glimpse of crotchety Carl Woese. Also, in the introduction to this book, I sketch what are going to be sort of the 3 main threads of it, and I mentioned that the book encompasses three big surprises. I think they're big surprises about who we are, we humans, we multicellular creatures, we complex creatures. 3 surprises about who we are and what we are and how life on this planet has evolved. 3 surprises. I'll read just one paragraph. And some of this you've already heard me sketching out. One of those 3 surprises involves an anomalous form of creature, a whole category of life, previously unsuspected and now known as the Archaea. That discovery in 1977. Another is a mode of hereditary change that was also unsuspected, now called horizontal gene transfer. The third is a revelation, or anyway, a strong likelihood about our own deepest ancestry. We ourselves, we humans, probably come from creatures that, as recently as 40 years ago, were unknown to exist. And what I'm alluding to there is what I describe at the end of the book, which is one of the discoveries that has come from the people who followed Carl Woese, and that is that our deepest ancestors are the Archaea. That there are, in a sense, only two major limbs off the tree of life, instead of the three that became orthodoxy after Carl Woese proposed it. And neither of those two major limbs is us. We are a minor limb, we eukaryotes, we animals, plants, fungi, complex creatures of all sorts. We are a secondary branch off of the branch Archaea, these creatures that nobody even knew they existed 40 years ago. And when they were first discovered, they were thought to be microbial bugs that lived only in extreme environments. The hot pots of Yellowstone National Park, salt flats, highly acidic environments, extremophile environments. And now we understand with a high degree of confidence, although some scientists are still arguing about it, that those are our deepest ancestors. Anyway, I wish if we were in a dark room and I were showing slides, I would be showing you pictures of Carl Woese, and I would also be showing you some crazy tangled trees and other pictorial representations of the history of life that Ford Doolittle, I mentioned him earlier, and other people have created to try and capture what we now see as this very tangled 4 billion year history of life. So why don't I pause there?
Okay.
And see if anybody wants to throw a tomato of outrage or.
Okay. Well, you were up a little earlier than I was, but anyway. Well, mine is kind of a basic question, so maybe it's good to go first. Just if you could fill in a little bit. First, I enjoyed it very much, everything that you had to say, and I have some background in all of the things you talked about, but not quite enough. And I'm wondering about the connection between two of the three threads. I'm assuming that the connection between the eukaryotes and the Archaea is horizontal gene transfer.
The eukaryotes and the Archaea?
No, the eukaryotes and Archaea.
You mean the non-eukaryotes?
The
bacteria. Yeah. No, the connection deep down is, at least as far as it's been plumbed so far, is a divergence that looks like a simple two limbs coming from a family tree.
But not as a result of horizontal gene transfer.
Later, there's horizontal gene transfer between them, but when they diverged originally, they wouldn't diverge by horizontal gene transfer. They apparently diverged by
normal means.
By normal, point mutation and selection. And some people have gotten alarmed when I talk later in the book, and I've talked in some other venues about how Woese felt toward Darwin. We can talk about Darwin and Woese and Darwin, but Woese's discovery and the discoveries that have followed that present, I think, important modifications to classic 20th century neo-Darwinian theory, but they don't overturn it. There's still incremental mutation. There is still natural selection, and there are these other factors too. Right. Anyway, go ahead.
Well, that's it pretty much.
Okay.
I just wondered the connection between the thread of horizontal gene transfer and his discovery, Woese's discovery of the eukaryotes. I mean, of the Archaea. So.
Yeah, both of these factors are happening. Incremental, slow motion, gradual mutation followed by natural selection, causing lineages to diverge, and they still certainly do diverge, including at the very base of the limbs of the tree, and horizontal gene transfer, which is infrequent, but over the long stretches of time has been abundant and highly significant.
I first was introduced to your writing in Ranamafan in Madagascar,
Oh, yeah.
where you had apparently gone to research with Pat Wright.
Yeah.
And there was your book, the Song of the Dodo, was there all covered with mold. And
Oh, you were in Ranamafan.
yeah, I wrote a book with about Pat Wright's work.
Oh, yeah.
Yeah. But what struck me about your writing was it was like a braided stream. And I think the way you're discussing this book, it's the way you're describing this gene transfer, it's really like an allegory to your writing, with the way you go kind of in and out of, and then come around and come back. But so I wanted you to comment on that.
Yeah,
but also, Lynn Margulis is
she's an important character in this book.
She is. Okay, that's what I wanted to know, because I know at UMass she was trying so hard to just get this accepted by the scientific community.
Let me say a word for everybody about Lynn Margulis.
And there's a book, a movie that just came out called Symbiotic Earth about her life.
I've heard about it, but I haven't seen it yet. So Lynn Margulis, a wonderful, brilliant, provocative, forceful, controversial microbiologist who died a few years ago, unfortunately, too young, and who spent most of her career promulgating a theory called endosymbiosis, which is a theory of the origin of the eukaryotic cell, these complex cells that compose our bodies and all animals, plants, fungi, complex creatures. Eukaryotic cells. Eukaryote meaning true kernel. They have cell nuclei, and they also have internal structures, organelles of various different sorts, just the way we have heart, lungs, liver in our bodies. Complex cells have organelles. Lynn Margulis revived a theory and revived elements of theory and combined them that had been proposed in the late 19th, early 20th century by some very interesting, fringy scientists, and she put those together into this theory called endosymbiosis, which asserted that complex cells have become complex and contain these important organelles like chloroplasts in plants that do the photosynthesis, and mitochondria in animals, which package energy. Hugely important organelles. She argued those are captured bacteria. Those are the descendants of bacteria that were swallowed early on in the history of life and became internalized and functional and adapted to be organelles. And people said, oh, she's wacky. She's nuts. For years. She suggested this in 1967. And then I've already mentioned Ford Doolittle. He was acquainted with Woese, and Woese was starting the sequencing. And then Ford Doolittle in the late 70s said, Lynn's theory is interesting, not necessarily crazy. Why don't we test it with genome sequencing using Woese's technique? And let's look at mitochondria, and let's look at chloroplasts in plants, because they have DNA in them too. Look, these mitochondria, they have DNA. Let's sequence it. These chloroplasts, they have DNA. Let's sequence it. He sequenced it, and what did he find? Matches with bacteria. Cyanobacteria for chloroplasts and a group called alpha proteobacteria for mitochondria. So he helped confirm what Lynn Margulis had been considered crazy for having proposed. And she's an important character in this book, and she's a great scientific figure. Okay, thanks for asking that.
I studied Japanese scientists who've been candidates for the Nobel Prize in physics and chemistry and medicine. And I can well imagine that Carl Woese and his achievements posed a major problem for Nobel committees in terms of what they would do, if anything.
Yeah.
So I don't think he ever received a Nobel Prize. There are a lot of important people who didn't. But I would like to know what kind of accolades he did get. Clearly he was a person who repelled some people and I suppose attracted a few friends. But was he a member of the National Academy of Sciences?
This is an important part of the story. Yeah, thank you. That leads to a couple of things. He was elected a member of the National Academy of Sciences, but at the age of 60, at which point he was frustrated that his colleagues had waited too long. Lynn Margulis got in at the age of 40, I think, or 45. He did not like that. They were rivals. He did not like her. So he was ticked off about that. He got a MacArthur Fellowship. He got the Crawford Prize from the Swedish Academy, which is considered the Nobel Prize for biologists. He didn't get the Nobel. Watson and Crick were biologists. Barbara McClintock was a biologist.
I was going to mention about McClintock. Her story reminds me a bit of what you said about Margulis.
Yeah, Barbara McClintock. And they're characters in this book too. Barbara McClintock. Crick is an important character. Watson, not so much. Crick was a friend of Woese's and helped Woese. But so there were people who were nominating Woese during his life for a Nobel Prize. And there were people who really thought that he should get one. Not least of all himself. But he didn't.
Okay. Thank you.
You're welcome.
So this may go into your promised discussion about Darwin, but I'd like to know your thoughts about what's happening to the old theory of competition red in tooth and claw, and now the new idea that maybe there's biological altruism and the trees communicate with each other to cooperate and protect each other. Yeah. And what does horizontal gene transfer
and cooperation and symbiosis, right?
Thank you.
Yeah. So yes. So that's a huge subject, an important subject, and I can just sort of dab at a couple aspects of it. Symbiosis was a big theme for Lynn Margulis, and not just this endosymbiosis as the origin of the eukaryotic cell, but the idea, and I've seen her lecture about this, and she's written about it, and a lot of other people saw her lecture about it. She firmly believed that in classic neo-Darwinian evolutionary theory, there was much too much emphasis on competition, struggle, strife, testosterone. You can sort of fill in all the blanks. This was kind of a male paradigm, I think, as far as Lynn was concerned. But she was not one-dimensional or tendentious. She was a scientist who was not trying to advance a feminist agenda, although she certainly was a feminist, but was trying to understand biology. She thought that competition was overrated and cooperation, symbiosis, was underrated. Now, that's not to be confused with the debate over what are the evolutionary origins of altruism, which involves Ed Wilson, and a lot of other scientists, and is still a very hot topic. And I'm looking into that now, not for another book project, but because I'm doing a different kind of project on Ed Wilson and his work. And I'm just starting to study up on that. His views have changed on that a little bit over the years. And have a lot to do with the origin of social behavior in ants and termites, as well as in humans and certain other mammals. Altruism. I won't try and summarize his views on that. Anyway, these things are all at play. There are a lot of really valuable, complicated, important, I'm not going to call them arguments, but discussions that are going on and need to go on within the community of evolutionary biologists. And of course, every time an evolutionary biologist says, well, Darwin was wrong about this, or these evolutionary biologists are wrong about that, then creationists will say, oh, see, they don't have their story straight, and this is just a theory. It's not that. It's the way a really complicated, robust science is supposed to evolve.
So I know much less about this than some of these people who've asked questions. But you talked about Archaea, that was, a third branch that we never heard of that's closely related to us. What happens in between? Does everybody go back and forth, in and out, and then we come up? it must have happened in a different way, or it happened by gene transfer. But how did we get from, we know how the eukaryotes developed, sort of, through DNA. I mean, we know through DNA we can sort of follow which way things go. But after the Archaea, what happens?
Well, Archaea probably haven't changed much in 3.5 billion years. Mm-hmm. We don't know that for sure because we don't have fossils.
Right.
We just have their DNA because they still live. They still exist on Earth. But inferentially, they are a primitive. They are a primitive, and primitive always has to be in quotes because they still do a really good job of being Archaea. But
right.
Relatively uncomplex. They still exist and they're thriving in their habitats. Meanwhile, we have diverged, it seems, from them, and with them as a container cell, the lineage that led to eukaryotes seems to have captured bacteria either by swallowing them or becoming infected with them. And with the mitochondria, this important kind of organelle that's in all of us packaging energy, it happened apparently just one time in the history of life. One archaeal cell internalized one bacterial particle, and that is the answer. You know, you talk about mitochondrial Eve, and there, you know, there's one woman who is the mother of us all, and that's mitochondrial Eve. Well, this is way before mitochondrial Eve. This is the one bacterial particle that got inside one archaeal host cell and replicated and evolved and changed and became all mitochondria in all complex creatures all over the planet. That happened. Plants got chloroplasts, also by bacterial capture, probably not just one time, but several times. And then complex cells evolved. And there's a big debate over whether eukaryotic cells, these ones that include us, whether they were really starting to be complex before they captured that first mitochondrion, or capturing that allowed them to become complex. There's a wonderful guy in Germany named Bill Martin who, if he were standing here and Ford Doolittle were standing here, they would argue each other into the ground on that question. And we would all sit here with our mouths hanging open wondering what the hell they were talking about. So it's complex, but that's at least does that answer it a little bit?
Sure.
Okay.
Thank you.
You're welcome.
Hi,
yes.
David. I actually worked in Ford Doolittle's lab at the time when he was writing those papers you were talking about.
What's your name?
My name's Arlen Stoltz. I didn't work on the phylogeny aspect, but
closer to the mic.
A little louder.
But in that group,
he worked in Ford Doolittle's lab during this crucial period of time.
Yeah, we all shared ideas with each other, and I've been trying to formulate a question, but things look completely different from my perspective, from the way that you've presented things. We didn't think of ourselves as Carl Woese's followers. We mocked Carl Woese's followers because he had followers. He had one of his talents was to inspire fanaticism, basically, about a particular method and about certain fixed ideas that he had about early evolution. So,
how about if I said Carl Woese's successors instead of followers?
Numerical taxonomy precedes Carl Woese and has completely different
Okay,
origins.
but how about this? I mean, I've talked with Ford about this for hours and hours and hours, and one of the things that's I don't want to cut you off. I want you, but let me throw this in.
Go ahead.
Linda Bonin, Carl Woese's right-hand person, lab assistant, who was helping him with this early method of sequencing in the 70s, moves to Halifax. She walks into Ford Doolittle's lab and says, "Any chance you have a job?" And he says, "Well, what can you do?" And she says, "Well, I was sequencing genomes with Carl Woese." And he said, "Great, let's do that." Right?
Yeah, he invented this absolutely fantastically valuable technique because you could do the same thing with every single species on Earth. You could get the same piece of information and then you could compare it.
Yeah.
Nobody had that before. That was the breakthrough.
Yeah.
But in terms of him discovering lateral transfer, that's
Oh no, okay.
that's what you implied.
Okay, I'm sorry I implied that. Yeah, Carl Woese did not discover lateral gene transfer. Yeah.
Yeah. So if I could
Horizontal gene transfer.
frame it as a question, I guess I would say it feels to me like your narrative. I mean, I'm sure all the facts are right in your book. The narrative framing is what I find disturbing, and I'm just wondering. It seems like this great underappreciated man theory of science. The maverick who's not appreciated, and that's the way science works. And my question is, what other narratives could you sell to an editor other than that one?
It's not just a matter of selling it to an editor. It's a matter of
that's
selling.
Not the narrative I would have used.
It's a matter of selling it to an audience of 60 people in an hour on a hot summer night.
Okay. All right.
I mean, if you read the book and feel like I've got it all wrong, please email me and tell me, and I will take that very seriously.
Facts. I'm sure you got the facts.
But what I'm giving you here is a cartoon version of the book that I've written.
Okay. All right. Thanks.
You're welcome. Thank you. Anybody else? Yes.
So I have a question that probably is a little bit of a follow-on, but first of all, I'd like to say, as a recent college graduate, I read Song of the Dodo and wrote you a letter, and you wrote me a very nice note back. So,
good.
So thank you for that. I've been a fan for a long time, and I was actually interested in maybe seeing if you would tie those two books together a little bit. That one is a bunch about Darwin and Wallace, but there are also more modern scientists who, in my now hazy recollection, were also somewhat iconoclastic, unappreciated, and that was part of your work. So I wonder if you have, from that work, a view of that is part of what's important to scientific discovery, those iconoclastic tendencies, or those are the people you like to write about?
Let me put it this way. People like to read about people. I'm acutely aware of that. And I'm very interested in science, but I understand that to write about science, you can't just explain it. You have to animate it. And the best way to animate it is by telling human stories. And if I have overemphasized in speaking tonight what is it, Arlen? Is it? Yeah, what Arlen said, which is very valid. I have emphasized two people, and I've mentioned a third. Carl Woese, Lynn Margulis, Ford Doolittle. Three very interesting people. There's a lot of scientists in here, and doing important work and who have interesting stories, and not all of them are outsiders, are thwarted geniuses. Joshua Lederberg, not an outsider or a thwarted genius. Francis Crick, not an outsider or thwarted genius. So there are lots of different narratives that show how science works. Science is a fundamentally human it is a human activity, and that means that it involves bioinformatics and genome sequencing, and also it involves a lot of coffee and a fair amount of argument and a fair amount of beer, and it involves people going to meetings and standing up and presenting papers, but it also involves people talking in the corridors of the hotels in which those meetings are held. It involves all of that, and I tried to get a lot of that into this book to animate the molecular phylogenetics, and I promise you, Arlen, that the book is not simply, here was Carl Woese and he was a thwarted genius and he should have gotten the Nobel Prize, even though I've emphasized that tonight. There's a lot of Ford, and there are a lot of other models of how you be a scientist and how you have a scientific career and how science works. Jim Brown, who you may have known from Ford's lab, has got an interesting story in here too. Yeah. Yes.
Yes, thank you very much. Could you please comment on what you've learned given the tendency towards genetically modified organisms and how this might play out?
Yeah. What I've learned is that it's been going on for 3.8 billion years. We have a tendency to say, oh, genetically modified organisms, moving genes from jellyfish into tomatoes, it's unnatural. Now, it can be, it's powerful technology, and there are things that can go wrong if you do it, and we need to take it very seriously. But it's not unnatural for genes to go from one species into another. That's one of the things that I've learned from studying molecular phylogenetics. It's been happening forever. Gary.
Yeah, you mentioned that 8% of the human genome is viral.
Did I mention that here, or was that?
No, it's in the book.
Yeah.
And,
okay, we were talking about this with Barbara.
Earlier. Is there any indication that that's likely to increase over time?
Let me back up and bring them into the picture on this. So another aspect of this is that in terms of the discoveries that have been made, 8%, we now know from human genome sequencing, 8% of the human genome is captured viral DNA. DNA, stretches of DNA in all of our genomes that do not come down by linear descent through the entire vertebrate and then animal lineage to us, but that have entered sideways over the last 10 million, 20 million, 60 million years, sideways by viral infection. Captured retrovirus DNA. Now, retrovirus, why do they call it a retrovirus? HIV is a retrovirus. Why do they call it a retrovirus? Because it goes backwards, retro, and it patches its DNA into the genome inside the nucleus in the cell that it infects. HIV infects immune cells, and it patches itself into the DNA of the immune cells, and then when an immune cell replicates, it also replicates the viral DNA, and then that cell explodes and out come viral particles, right? That's HIV. If a retrovirus infects, the cells in an ovary or testes or a sperm cell or an egg cell, the reproductive cells of a particular creature, then it becomes heritable. Then it gets passed on and can potentially become, if that creature has offspring and a long lineage of descendants, then it can eventually become part of the lineage. Well, 8% of our genomes have been acquired that way, including, this is a story I tell at the end of the book, including one stretch of DNA that has been identified as a functional gene, formerly a viral gene that formed an envelope around viral particles. They call it the envelope gene, but in mammals has been adapted, changed, evolved, adapted to form a membrane, kind of an envelope, between the placenta and the fetus. And without that membrane, pregnancy is not possible in mammals, including humans. That's a viral gene. Co-opted, repurposed to make pregnancy possible in mammals. I forget what the...
Yeah.
Oh, is it still happening? Is it still happening? Well, there's no reason to assume that it's done forever, but insofar as it's been detected so far by this wonderful scientist named Thierry Heidmann in Paris that I went to see. He's a character at the end of the book. It happens infrequently that a viral gene is internalized and perpetuated and stays within the lineage, including the human lineage. So we know that some of these have come in. There are different versions in different mammals, suggesting that it has happened at different times in the course of the evolution of mammals, and that maybe the one that we have in humans is an improved version that's substituted for the one that our mammal ancestors had 60 million years ago. So it's still happening if you count the last 20 million years as now. Okay?
Okay, Okay, we're beginning to run out of time, so I'm just saying this is the last question.
Okay, last question.
Okay, thanks, David. I haven't read your book yet, so I can say this freely, but the best science book I've ever read was by Nick Lane called The Cell. And I measure how much I like a book by how many notes I make in pencil in the back of the book. And I think I had 2 pages of tiny little notations. And Nick basically goes through the history of life. Of course, this was like 10 years ago. But if anyone wants to follow up on this field, Nick Lane's The Cell is just an amazing book.
Okay, also The Vital Question by Nick Lane.
Later.
Yeah. Nick Lane is a very, very smart, interesting. He's a scientist who manages to write well too.
Right. He's highly involved in the field of deciphering the history of life.
Yeah. Okay. With that, we'll tip our hats to Nick Lane, and I thank you all.
Thank you.
Thank you.